All right, so welcome back from spring break. Before we jump into chapter 12, I want to say a little bit about the new format of lectures. So a lot of people reached out to me and said, please try and record your lectures so that we don't have to go to PowerPoint, because I think people like the whiteboard method. So that's what I'm going to try to do. I haven't changed my lecture notes at all, but one thing that's going to be different is that I'm not going to be able to jump back and forth between the whiteboard and the PowerPoint, because I only have room with my little camera for just the whiteboard. I can't fit the PowerPoint in there as well. But hopefully what you're doing is you're watching this on a laptop. So if I switch over to start talking about the PowerPoint, then you can just minimize the video and open up the PowerPoint. So you can have them both open concurrently. And if I walk off the screen over there, then what I'm doing is I'm walking over to that computer so that I can advance to the next slide and see the PowerPoint that I'm talking about. So yeah, if you've got a laptop or a desktop computer, that's the way to do it. If you don't have a laptop or a desktop computer, if you're doing this on your phone, it's going to be pretty difficult. But if that's the case, then shoot me an email and we have ways around this. So if you only have access to a phone and not a computer, then let me know and we'll get you whatever, we'll do whatever we got to do. These lectures are going to go a little bit faster because I'm not going to stop and ask questions, right? And I don't need to slow down now because we got a video, right? I don't need to slow down so that people can keep up with their notes, right? If I say something that makes the light bulb switch on for you and you need to write down notes and do whatever you got to do, you can just hit pause, and then when you're all caught up, jump back in with us. And so these will go a little bit quicker, but I strongly encourage you to pause the video a lot, replay things. If I say something and it doesn't click with you, maybe listen to it again. And so take advantage of that fact that it's a recording. You can pause and rewind and listen to things over again. So this might actually help a lot of you be able to really get all of the information and not get behind during lecture, because I know that sometimes that's an issue with some people not being able to keep up with lectures. What else? That's it. All right, so on to chapter 12, the cell cycle. We're not going to do the rest of chapter 10. It's a shame. It's one of my favorite chapters, chapter 10. But sacrifices have to be made. Also, I'm going to try and walk up and talk at the camera a whole lot, because I realize if I turn this way and talk, then you can't really hear me. Okay, so chapter 10 is all about the cell cycle. So this is just the cycle that cells go through when they're replicating and dividing. So when people first started to look at cells under microscopes, what they noticed is that some cells would go through this phase where they dramatically change their appearance, and then they split into two cells. And so when people were first looking at cells under microscopes, they didn't really know what exactly was happening. All they saw were cells that were dividing and cells that were not dividing. And so as far as they could tell, there were only two phases that were things that you could describe. And so they named the cell division phase mitosis, and then everything between mitosis just got lumped under interphase. So an early explanation of the cell cycle looked something like this. Right? If this circle right here represents a clock that the cell just goes through, this part right here represents mitosis, and then everything else they just considered not mitosis, or the name for it is interphase. So you have mitosis, which is cell division, and then interphase, which is not cell division. Right, and so this cell will just proceed clockwise around this cell cycle, going through phases of mitosis and then not mitosis. And this is a very primitive explanation of what's actually happening, because there's really there's a lot going on here in interphase. It's not just downtime, right? There's a lot of stuff going on in here that's either getting ready for mitosis or just doing all the things that that cell does in its regular day-to-day activities. So, once we started, once scientists got better at really looking at what was going on inside the cell, they realized that there's really more to it than just mitosis and interphase. So interphase can actually be broken up into 3 separate phases called G1, S phase, and G2 phase. So first the cell will go through G1, and we'll talk about what goes into each one of these phases in a little bit more detail. S phase, and then G2 phase. So the cycle that the cell goes through that we understand now is that it goes through mitosis, which is cell division, then G1, G stands for gap, right? It's the gap between mitosis and DNA replication, right? So you have mitosis, which is cell division, and then a gap, and then S phase where the DNA is replicated, and then another gap. So mitosis or M phase, G1, S, and then G2, and then you're back to mitosis again. So the cell just kind of goes through this cycle of dividing, taking a gap, and then replicating its DNA, and then another gap, and then dividing again, and it goes along like this until it just stops dividing. So before we go into what actually happens during each one of these phases, go ahead and label M here. Before we go into what happens during each one of these phases, we'll make a checklist of things that need to happen before a cell can divide. So this is our cell division checklist. So this is in no particular order, right? Obviously, the cell has to make a copy of all of its DNA. All right, that's one thing that has to happen. All right, so the cell has to make a second copy of DNA. the chromatin, which is the actual DNA, it has to be condensed before it gets divided. So, you know, when you look at the DNA inside of a nucleus that's not, you know, actively dividing or getting ready to divide, the DNA is just in there all kind of loosey-goosey, like a bunch of like loose spaghetti, right? So before the DNA can be organized and separated, it has to be condensed. So the chromatin has to condense, and then the DNA gets separated into two separate daughter cells. So mitosis produces two daughter cells, and each one gets an equal and, you know, in a perfect world, an identical set of DNA. But it's not just about replicating DNA and splitting up the DNA. Another thing that has to happen is all those organelles. You got to make enough organelles to support both of the new daughter cells. So the cell has to make a bunch of extra organelles, and it has to also make a bunch of extra membranes, because each cell needs its own set of membranes. So we need extra organelles, and we need extra membranes. So obviously this stuff needs to happen in a particular order, right? The DNA copies can't be distributed before the chromatin condenses, and it obviously can't be distributed before a second copy of the DNA is made. And cell division isn't over until all of this stuff has been divided up between the two daughter cells. So we'll start with S phase. We'll go into each phase and look at what happens in each phase. And we're going to start with S phase, because if S phase happens, right, S is when the DNA gets replicated. If S phase happens, then generally the cell will continue all the way through the cell cycle until it gets back to G1. So this is actually a good place to start when you're looking at cell division. A lot of cells, they just stay in G1. This is the gap phase. They'll stay in G1 indefinitely until there's some trigger that causes cell division. And when that trigger happens, they'll enter S phase, and then once they enter S phase, they'll continue along until they get back to G1, and they'll stay there until there's some trigger that causes them to enter S phase again. So that's why it's a good place to start. So in S phase, there's really only one thing that happens. The DNA gets copied. I mean, there's more stuff that happens, but for the purpose of Biology 1107, all you need to know is that in S phase, the DNA gets copied. And we'll go into the nuts and bolts of how that actually happens in chapter 16. We'll color code it. So after S phase, then the cell enters G2 phase. And so G2 phase is just the period of time between DNA replication and cell division. So in G2 phase, the DNA has already been replicated. So the cell is sitting there with 2 copies of each chromosome, and it's just getting ready for cell division. So during G2 phase, there's a whole lot of metabolic activity. This is when the cell really ramps up its membrane production. So you see usually very large endoplasmic reticulum, a lot of protein being made, a lot of organelles being replicated, because the cell is getting ready to make another copy of itself, and it needs all of that other extra stuff, not just the DNA. So yeah, here you see a lot of metabolic activity. And so once G2 phase is over, then the cell will enter M phase, or mitosis. So this is cell division, right? We've talked about this a little bit. But basically during mitosis, you have all of that stuff, the cytoplasm, the organelles, the DNA, all that stuff needs to be divided up between the 2 cells. So that's what happens during mitosis, or M phase. So you got your cytoplasm, your organelles, and your DNA all need to be divided up between the 2 daughter cells. And then you're back in G1 phase, right? And so you're back at the beginning. And the cells in G1 phase, they're really not doing anything with regards to cell division. They're not getting ready for cell division. They can stay in here indefinitely. And they can stay in here until something triggers them to jump back into the cycle, replicate their DNA, get ready to divide, and then get back to G1. So this is just your regular day-to-day cell activities. Depending on what kind of cell it is, it's just doing whatever that cell does. And it's not really getting ready to divide or anything like that. So if sorry, I stopped talking when I turned back this way, because when I got my face pointed at the board, then the video goes really quiet because my camera is not that great. Okay, so if a cell exits the cell cycle at G1 and just hangs out in that indefinite G1 phase, then we say that the cell is not in G1, it's actually in G0 phase. So G0 phase is that indefinite G1 phase. And the cell can stay in G0 phase, for the rest of its life. Okay. So that's a brief introduction to the cell cycle. And what we're going to do now is we're going to go into all the different stages of mitosis. So what we were looking at there is the phases of the cell cycle. M phase, G1, S, and then G2. Now we're going to focus a little bit more on M phase, mitosis. We're going to focus on mitosis and look at the different stages of mitosis. And we're going to look at how the DNA, or how the cell actually executes that cell division event. So before we jump into that, I'm going to run out of batteries. Before we jump into that, I want to talk a little bit about how the DNA is actually organized. So in a eukaryotic cell, the DNA is packaged into a structure called a chromosome. All right, a chromosome is just a linear strand of DNA that can have, depending on the chromosome, it can have hundreds of genes or it can have thousands of genes. So this is in a eukaryotic cell. So we're not talking about bacteria. We're not talking about prokaryotes, because prokaryotes actually have that circular DNA strand that we were talking about. These are not linear. Chromosomes are linear. And I'm actually going to pause the video here because I need to plug in. And we're back. All right, so what I want to do now is look at the organization of these chromosomes in the cell as the cell goes through the cell cycle. So if you got a cell, all right, let's say this particular cell only has one chromosome. We'll draw a chromosome like that. So that red thing is a chromosome. And here, this particular cell only has one chromosome. It's the easiest way to conceptualize what's happening to the chromosomes as the cell goes through the cycle. So right now, the cell is in G1 phase. All right, so the cell's in G1 phase, just carrying on with its day-to-day activities. And then there's some trigger that causes the cell to divide. The first thing the cell's got to do is it's got to replicate its DNA. So it will go through S phase. And once it goes through S phase, if you remember, what happened in S phase is the DNA was replicated. So now we don't have one copy of the chromosome. We have two copies of the chromosome. And so these two identical copies of the chromosome are called sister chromatids. Each of these is a chromatid. These are sister chromatids. And they're just identical copies of the same chromosome. So that's what the cell's got in G2 phase. And then the cell goes through mitosis, or M phase. And then after mitosis, the cell has divided into two daughter cells. Each one has one of those chromatids. And so now the cells are back in G1. So these are what I keep referring to as the daughter cells. All right, so now what I'm going to do is, we've kind of beat this stuff to death. What I want to focus on now is this M phase, mitosis, because mitosis has a number of steps that are pretty well characterized. So we're going to focus in on what actually is happening during mitosis. So when the cell gets ready to divide, the DNA condenses into those chromatids. So when the cell is just, in G2 phase, and it's had its DNA replicated and it's getting ready to divide, the DNA is still pretty loosely organized in that, pile of spaghetti looking arrangement that we call chromatin, right? Chromatin, T-I-N. When the cell gets ready to divide, then all that chromatin condenses, and the chromosomes will condense into those sister chromatids. So this is a structure that you've probably seen before. You probably saw it in high school biology, right? Where you have a chromosome that's like this, and then its sister chromatid right there. So these are your sister chromatids. I got a chromatid here and a chromatid here. And the chromatids, don't confuse it with chromatin, two different things. The chromatids are held together along their entire length with proteins called cohesins. So that's what I'm drawing in red there. These are proteins called cohesins that bind the sister chromatids together along their entire length. And then there's one area in the middle, right, where the two chromatids are very tightly bound together. Called the centromere. And so now if you look at slide 1, if you look at the very first slide on the PowerPoint, what you'll see, I'll give you a second to minimize this video, open up the PowerPoint. We're done. All right, it's open. You're looking at the PowerPoint slide 1. What you're looking at there at the top says sister chromatids. What the arrows are pointing at, the green arrows are pointing at the centromeres. So you can see each one of those structures that looks kind of like an X, right? Some X's are bigger than others. Each of those X-looking structures is a pair of sister chromatids, and that area in the center of each one, where they're very tightly bound together with that green arrow on it, those are the centromeres. So they're very tightly held together at the centromere, and they're loosely held together along their entire length by the cohesins. So this is the first step when the cell gets ready to divide, right? When mitosis happens, this is the very first thing that happens. And this is hard. I keep wanting to say, is it okay if I erase it? It's almost like I don't even think about it when I say that. Okay. So we'll talk about the different phases of mitosis now. Now, when I was your age, we only had prophase, metaphase, anaphase, and telophase, but you're lucky, you get 5 phases. So you get prophase, prometaphase, metaphase, anaphase, and telophase. And so we'll talk about what goes in each one of these phases. But I'll write them on the board real quick, just so you can have them down in the correct order. They go prophase, prometaphase, metaphase, anaphase, and telophase. And so we'll start with the obvious choice, prophase. The thing that happens during prophase is what I just wrote on the board when I was drawing out the chromatin condensing into the chromatids. That's what happens in prophase. So in prophase, the chromosomes condense into those sister chromatids that you drew a second ago. And I'm kind of crooked here if we do that. Okay. Yeah. Another thing that happens is the mitotic spindle forms. That's a new one for you. The mitotic spindle forms. The mitotic spindle is basically just a microtubule structure that provides the scaffolding for pushing the replicated chromosomes apart to opposite ends of the cell, right? So you remember how we were looking at the microtubules as this intracellular superhighway, right? And we watched that video of the kinesin protein walking along a microtubule and carrying a big vesicle behind it, right? So that's what the mitotic spindle is. It's this big network of microtubules that provides scaffolding for the chromosomes to be pushed apart into the 2 new cells. And so it just kind of looks like this. You've got a centromere on one end. Remember, a centromere is just kind of like 2 bundles of microtubules sitting at a right angle. So you'll have a centromere at one end and a centromere at another end. And then microtubules extending between them. So these microtubules will polymerize and push the chromosomes apart. And so now if you'll advance to the next slide, this is slide 3, you'll see 3 pictures labeled interphase, prophase, and prometaphase. And so what you're looking at now is you're looking at a cell, obviously, that's in each one of these phases. One in interphase, one in prophase, and one in prometaphase. And the one in interphase is just, just like a regular cell. But if you look at the one in prophase, what you'll see is that the blue, the nucleus in the middle, right? The thing that's staining blue is the DNA. And in interphase, the DNA is fairly dispersed, and you see this big uniform blob of DNA in interphase. But in prophase, what you'll see is it looks like the DNA is starting to take on this kind of wormy appearance. And that's because the chromosomes are condensing and they're forming those, pretty dense structures, the chromatids. And so you can see that in prophase. You can see that starting to happen. And you can also see the mitotic spindle starting to form. The microtubules in these images that you're looking at, the microtubules are stained fluorescent green, so that that green starburst looking structure, that's actually the one of the centromeres starting to form, and the microtubules extending off from that centromere. So yeah, that middle picture, that's prophase. Right? You can see the chromosomes starting to condense, and you can see the mitotic spindle starting to form. It's that green structure. Next phase. Prometaphase. In prometaphase, one big thing happens. It's the nuclear envelope falls apart. So you remember when you were looking at the structure of the nucleus, and it has that dual membrane that encloses all the DNA. In prometaphase, that whole thing falls apart. So if you look at that picture all the way on the right, for prometaphase, now the DNA is not all held tightly within the nucleus. The nuclear envelope is completely fallen apart, and so now the DNA kind of fills up almost the entire area of the cell. And so that's what you're looking at there in prometaphase. And in prometaphase, once that nuclear envelope falls apart and the replicated chromosomes kind of spread out a little bit, then you can really start to see individual chromatids. And so it's a little bit easier to see those structures in prometaphase. And one thing you can see in that particular prometaphase, you can see that the mitotic spindle is now clearly divided in two, right? You have a centromere at the top and you have a centromere at the bottom in that picture. And so that's your mitotic spindle, and the chromosomes are starting to attach to the mitotic spindle. So that's the other thing that happens in prometaphase, is the microtubules attach to the chromatids. So the microtubules will attach to the chromatids via proteins called kinetochores. Right? So if you look at the structure here, all right, here's our sister chromatids, right? There are proteins called kinetochores right here at the center that attach to the centromeres, and then the microtubules attached to that. So these kinetochore proteins just kind of act as a bridge between the microtubules and the chromatids. So let's see, the kinetochore, that's the green thing. And then in red, you're looking at microtubules. And these are chromatids. And I've got a picture of kinetochores. If you advance to the next slide. What you're looking at now is an electron microscope image of a kinetochore protein connecting microtubules to one of these chromatids. So on the left side of the image, that very dark staining, that really dense dark staining stuff, that's a chromatid. And then the big green arrow is actually pointing at the kinetochore protein. And then extending off the right of the kinetochore protein, you can see the microtubules just kind of extending straight off to the right of the kinetochore protein. So that's what they look like. And that's the end of prometaphase. So in metaphase, the chromosomes have already attached to the microtubules, and now they need to align themselves across the center of the cell. And that's what happens in metaphase. So in metaphase, the chromosomes will align themselves at what we call the metaphase plate. And the metaphase plate is not actually a physical structure. It's a plane along the midline of the cell in which the chromosomes get arranged. I'll show you a picture if that didn't make any sense. So the metaphase plate, I've actually got a, yeah. It's hard to tell in this picture. So if you advance one more slide where you're looking at three phases, you're looking at metaphase, anaphase, and telophase. If you look at the metaphase cell, what you'll see is that the cells are the chromosomes are all bunched up in the middle of the cell. So in metaphase, the chromosomes have all kind of migrated to the center of the cell. And if you look, right, I'll actually draw it with the same orientation that it has in your PowerPoint. If you look at a cell that's in metaphase, you'll have a centrosome on one end and a centrosome on the other end, and then microtubules running between them like this. And right about halfway between each of these centrosomes, that's where you find the chromosomes. So they just kind of arrange themselves in a plane that's roughly equidistant between the two ends of the cell. So this right here, well, these are your chromosomes in red. And the plane along which they align themselves is called the metaphase plate. So it's not actually a structure. It's an imaginary plane that's equidistant between the two centromeres. Centrosomes. All right, it's metaphase. anaphase. In anaphase, this is the beginning of when the chromosomes actually separate. So at the very beginning of anaphase, the cohesin proteins get cut. If you remember back earlier, we were talking about the structure of the chromatids. The cohesin proteins were those proteins that were holding the chromatids together along their entire length. So at the beginning of anaphase, those proteins get cut, and so now the chromatids are no longer held together. So now the chromatids separate. So in anaphase, the cohesin proteins get cut, and then the chromatids separate, and they're pulled towards opposite ends of the cell. And that's what you can see in the center image on this slide, the image labeled anaphase. You can see pretty clearly now that there's two bunches. There's two bunches of chromatids that are being pushed towards opposite ends of the cell. And so if we were going to draw that, if we were going to draw a cell in anaphase, you still got these centrosomes. But now, well, we'll do it like this. Now the microtubules don't necessarily extend all the way across, but they're shortening and bringing the now separated chromatids towards opposite ends of the cell, right? So the chromatids, the sister chromatids, are being pulled away from each other towards opposite ends of the cell. So that is anaphase. And then you've got telophase. And in telophase, the chromatids, the sister chromatids, have already been separated to opposite ends of the cell. And what needs to happen now is the nucleus will reform around those chromatids. So now you'll actually have two nuclei reform. One around one bunch of chromatids, and then the other one around the other bunch of chromatids. So the nuclei reform, the mitotic spindle will disappear. So the mitotic spindle disappears, and at this point, mitosis is complete, right? And you can think of mitosis as just the division of genetic information between the two cells, right? So once telophase is over and the nucleus has reformed around those two bundles of DNA, then mitosis is complete. But we still have one more thing we need to do. and that is cytokinesis. Cytokinesis is not a phase of mitosis. It's the event that actually pinches the cells apart, right? So this is something that actually can start happening during anaphase and extend throughout telophase. It's not an actual phase of mitosis, cytokinesis. It's an event that can span multiple phases. And what happens is there's a contractile ring of proteins that'll surround the cell like a belt, and it will tighten and tighten and tighten and tighten until it eventually pinches the cell in two. So you'll have this contractile ring of actin filaments that will pinch the cell in two. So if you're looking, this is a cell that's being pinched in two, right? So there will be a ring of actin that rings around and all the way around, and that ring will just get smaller and smaller and smaller until it pinches this cell in two. All right. So one last thing I want to talk about is how the cell cycle is actually regulated. So we'll talk about cell cycle regulation. It's not a thing that just continuously goes. I kind of alluded to this earlier when I was talking about G0 phase, right? The cell doesn't just constantly go through all these phases of the cell cycle. It'll stop at certain checkpoints where the cell will essentially check to see if certain conditions are met. And if all of the conditions are met, then the cell will progress on to the next phase. And so we call these checkpoints. So the cell cycle halts at checkpoints. And it will only proceed if specific conditions are met. Right? So I'm going to erase this, and we're going to put our cell cycle back on the board. So we're going to put, here's our cell cycle. Put that back on the board. Up here we've got M phase and we'll make it a little bit cleaner. All right, so we've got M phase, and then G1, and then S phase, and then G2. So, one checkpoint is between anaphase and telophase. So there's a checkpoint right here. There's a checkpoint during mitosis between anaphase and telophase. And if you think about it, it makes perfect sense that there would be a checkpoint there. If you think about what's actually happening in anaphase and telophase. In anaphase, the chromatids are being pushed apart to opposite ends of the cell. And then in telophase, you have a nucleus forming around each separate bundle of chromatids. So if you think about it, if that nucleus started to form before anaphase was complete, then you might run into some trouble, right? You have to fully separate the chromatids in anaphase and then check to make sure that that's done, right, at the checkpoint. And if everything is done, if anaphase is complete, then telophase begins and the nucleus begins to reform around the chromatids. So you have an M checkpoint between anaphase and telophase. You have an M checkpoint between anaphase and telophase. And there's also one between metaphase and anaphase. All right, there's another checkpoint between metaphase and anaphase, again, for obvious reasons, right? If you look at what's happening in metaphase, the chromosomes all align across the center of the cell, and then in anaphase, cohesins are clipped, and the chromatin separates, or the chromatids separate. So there's a checkpoint at the end of metaphase, and the cell will only clip those cohesin proteins if the chromosomes are all aligned perfectly along the center of the cell. It's just a way to keep everything organized. So you have these checkpoints that make sure that those conditions are met before the process can continue. Here's another one here. There's a G1 checkpoint. There's a G1 checkpoint. Most cells in your body actually never pass this checkpoint, right? Most cells in your body are not actively dividing. So if a cell gets to the G1 checkpoint and fails that checkpoint, that's when it will exit the cell cycle and enter G0 phase. So failing checkpoints is not always a bad thing, right? So the cells that fail this G1 checkpoint, they just go into G0 and they stop dividing and they just do whatever it is that that cell does. You got another checkpoint in S phase, right? This is an important one. It will stop the cell cycle if DNA damage is detected. So you have an S checkpoint, and at the S checkpoint, everything stops. The cell checks to see if there's any DNA damage, and if there is DNA damage, then it'll kill itself. I should say, if it detects any cell damage or DNA damage, it'll kill itself, because sometimes it goes undetected. But the DNA damage that does get detected here will stop the whole cycle and the cell will not replicate if it detects DNA damage at the S checkpoint. All right, so that's it for regulation of the cell cycle. I'm going to talk about the different proteins that actually execute all of these functions of the cell cycle. All of these activities that we've been talking about, you know, the microtubules polymerizing, all of the proteins getting replicated, well, not proteins, but you know, the chromosomes being aligned at the metaphase plate and then the cohesins being cut. All of that stuff is carried out, is executed by proteins called cyclin-dependent kinases. And so if you're keeping up with your vocabulary, you'll recognize this word, right? A kinase. An ACE, A-S-E, just implies it's an enzyme, right? It's a protein that catalyzes a chemical reaction. A kinase uses ATP to catalyze an energetically unfavorable reaction. So these are the proteins that do all of that stuff associated with the cell cycle. So yeah, they use ATP to do all of that stuff. And so you can just kind of think of them as mitosis effectors. They're the machines that do all of the stuff associated with mitosis. And you would think, right? You would think because the cell's not always going through mitosis, you might think, well, the cyclin-dependent kinases only get made when they're needed. The reality is that they're always there. So they are present at a constant concentration. They're always there in the cell, but they're not always active. They only get activated if the cell needs to go through mitosis. And they get activated by proteins called cyclins. They're only active when bound to cyclins. That's why they're called cyclin-dependent kinases, right? If they're not bound to a particular cyclin, which is another protein, if they're not bound to a cyclin, then they're not active. They're just hanging out in the cytoplasm, not doing anything at all. So that brings us to our next class of proteins that regulates this whole mess, which is cyclins. All right? So you got cyclin-dependent kinases, which are present all the time. Cyclins, on the other hand, have this cyclic expression. It's where the name comes from. Cyclins have cycles of expression. And they activate cyclin-dependent kinases. I should say CDK. Cyclin-dependent kinases, CDKs. And so yeah, the cyclins are called cyclins because they have cyclic expression patterns. And what that means is as the cell progresses through the cell cycle, it will activate or it will start producing different cyclins. And those cyclins will only be around for a set amount of time before they degrade. And then another set of cyclins is made. And so different CDKs will be activated depending on which cyclins are being produced by the cell. So that's the way this is all coordinated. And one last thing, I want to talk about a few different external factors that can regulate the cell cycle. One of the external factors that can regulate the cell cycle is a class of proteins called a growth factor. A growth factor is just a protein that's made by one cell that can promote division in a different cell. And I apologize if I'm writing small. I'm getting used to this. So a growth factor is just a protein that's secreted by one cell that can cause another cell to divide. Another external factor is called density-dependent inhibition. So a lot of cells, they'll just divide until they run out of room. They might divide until they start bumping into other cells and they get walled in and they run out of room, and so then they'll stop dividing. That's called density-dependent inhibition. All right, and then the last thing we'll talk about is anchorage dependence. This doesn't apply to all cells, but some cells require anchorage. They need to be holding on to something in order to keep dividing, right? And if they break loose from whatever tissue they're in and start floating around, then they'll stop dividing. No, they need to be grounded. Some cells. Not all cells have anchorage dependence. So cells that do have anchorage dependence need to be attached to a surface in order to keep dividing. All right, and that is the end of chapter 12. Go wash your hands.